For those who are newer to CMT and to our work at the foundation, I want to start with something that shapes everything that we do. CMT is a genetic disease. And because of that, many researchers and many of us in this community believe that the most powerful solutions will ultimately be genetic in nature. That belief drives our investment strategy in a real way. CMTRF aggressively funds a gene therapy portfolio, and we have an active investment in projects looking at 1A, 1B, 1J, 4J, 2A, 2E, all of the Rs. And some of these programs are in preclinical development. And we're meeting with companies that are exploring gene therapy approaches for all sorts of subtypes all the time. We're watching them very closely. Now, these therapies aren't without their complications, and they aren't without controversy. There's something we want to be very honest with you here tonight, but they offer a genuine, compelling approach, not just for CMT, but for many diseases, and the science is moving fast. Here's one example that I find remarkable. The FDA recently approved a genetic medicine for a special kind of inherited deafness called auditory neuropathy. And much like CMT, this form of deafness is caused by a well-characterized genetic mutation. One of those genes is called otoferlin. The approved treatment is called Otarmeni, and it's reaching patients right now and changing lives in real time. The scientific process behind that therapy was led by the person sitting with us here tonight, Dr. Meghan Drummond, spearheaded that drug development process at Regeneron. And now she's our VP of Research and Drug Development at CMTRF. And I cannot overstate how excited we are about the experience and the leadership that she brings to this work. Tonight, Meg is going to walk us through the fundamentals of gene therapy and what they mean for CMT. But before I hand it over to her, I want to say this. The foundation and the CMT research community are expanding her expertise into real progress for our community. And you're going to hear some of that progress tonight. We have a few important updates and announcements that we want to share with this group. But first, we want to have a genuine thank you for showing up here tonight. We'll get our announcements out after Meg's presentation, so please stick around. Now, before I fully hand it over to Meg, I want to prepare you for how she's going to open. You're not going to hear from her first. You're going to hear from someone whose life was changed by this science. Meg's presentation is going to begin with a short series of videos of one of the first patients to receive this gene therapy. I've seen it, and I'll just say that it puts everything into perspective. The baby in this video is Opal. Before receiving this therapy, Opal was completely deaf, blocking her ability to fully experience her world and ultimately develop speech. This work is not abstract. This is what we're working towards for CMT.
Great. Thank you so much, Laura. And here, let me turn my video on, though I'm not the person you want to see right now. So let me show you this video of when we first knew that the gene therapy worked in the very first patient dosed with the otoferlin gene therapy. So that was the first time that the mom had realized that her baby could hear. She didn't have her cochlear implant in, she had made a noise, and the baby, Opal, had turned toward the noise. And that was the moment that the mother knew that her child was no longer deaf. And now let's fast forward several months.
Yeah. They're called dungarees. Dungarees. T-shirt. What does it look like? Shorts. Good girl. Trousers. Coat. Scarf. Yeah. Cool. Pajamas. Good. Yeah. Vest. Yeah. What colour are the wellies? Good girl. What colour are the sandals?
Pink and red.
Good girl. Well done.
What colour is that?
They're red, aren't they?
What that?
Trainers. What colour are those? What colour are those?
I see, but it is on
Yeah, good girl. In the very distant noise, you can hear an ambulance. That'll be on the busy road. That was super good listening. What noise does the ambulance make?
the
Good girl. Can you say?
All right, now aside from the cuteness factor being incredibly high for that video, one of the things that's truly remarkable about this is that not only was the baby or the child communicating with her mother, right? Not only was Opal having this conversation with her mother, doing something very developmentally normal for that stage of her life, but also while having this conversation, she was sampling the environment around her, which is an advanced and complex form of hearing. So not only did we restore normal speech and language, but we also has restored these advanced metrics where we're in the far distance. She hears an ambulance and tells her mother while they're having a conversation, oh, wait, there's an ambulance over there, and then makes the noise of that ambulance. So, really, it was just truly remarkable and really a huge advancement for the field of gene therapies, and something that continues to give me hope, and why I'm excited to talk to you tonight about the future of genetic medicines for CMT. The goal of tonight's presentation is to walk you through some of the fundamentals of gene therapies and genetic medicines, give you some terminology to hold on to, and answer any questions that you may have, things that have been on your mind or things that maybe you thought about for the first time tonight. But to bring this and hopefully bring it to life and to so that you can appreciate these advancements that are in the very near future for many types of genetic disease, but especially for CMT. So with that, I'm going to now share my screen for my presentation. And feel free, as I pull this up, to, if you have any questions, put them in the chat, and we will answer them either during or at the end of the presentation. All right, and now let me share this, and I think we're all set. Okay. So I'd like to start with a little bit of foundational information to help understand and appreciate exactly how a genetic medicine works. So your body is built from trillions and trillions of cells. There's estimated to be about 30 trillion cells in the human body, and almost all of those cells, except for your red blood cells, have a nucleus, and inside of that nucleus are 23 pairs of chromosomes, and each of those chromosomes is made up of DNA. That DNA is wound very tightly and compacted, but if you were to unwind all of that, you'd find that it's made out of four nucleotides, or what scientists call base pairs, and we label them A, G, C, and T. And so A, G, C, and T are the alphabet of our genome, and that's what gives our cells instructions for how to function. The human genome, even though it's massive in size, initially was, as scientists began to sequence the human genome and decode it, it was estimated that there'd be hundreds of thousands of genes. And in fact, the community was surprised to find that there's really only 20,000 genes, but a lot of what's called regulatory and non-coding sequences that tell what genes when to turn on and how to turn on and how much of it should be made. And the fundamental way that this works is that you've got your DNA, which is like your library or your cookbook. You have a gene, which is one set of instructions or a recipe. You have your mRNA, which is the translated version of that gene. That's the instruction that's saying, here's exactly how we do this. That mRNA is translated into a protein, and that protein is what actually does the function, or it gets the job done. And so if we back all the way up, we go back to the instructions, the DNA, and the genes in the DNA that encode proteins that give you function. Now, one of the very cool things about human biology is that in every single body of our cell, again, except for those red blood cells, you have the same set of instructions. You have the same chromosomes, you have the same 220,000 genes. But what makes one cell different from another is when and how much of different genes that you turn on or off. So, for instance, a neuron and a heart cell both have the same instruction book. But a neuron is going to turn on genes that are specific to neuronal function. I've listed a few down here, similar to a muscle cell, which is going to be a slightly different set of instructions than a neuron, and a Schwann cell, which is going to again be different, and a heart cell. So each of these cells, the neuron, which is that sends electrical signals and messages throughout the body, the muscle cells, which generate force and movement for us, those Schwann cells, which insulate our nerves with a myelin sheath that allows us to send signals for very long distances throughout the body. And then, of course, your heart cell, which pumps rhythmically and allows blood to flow throughout your body. So the specialization really in how we are made and how we operate comes from selectivity of turning these genes on and off whenever they're needed to have a cell perform a particular function. And there's a lot of ways that this can go wrong. There's a lot of genetic forms of disease that occur when you have mutations of these genes. And CMT is one disease, right? So it's one disease that's a peripheral neuropathy, and that's what unites this diverse set of genetic underpinnings, is that you have progressive damage to the peripheral nerve, either from mutations that affect the function of the axon, which is shown here in this yellow color, or the Schwann cells, again, that provide that insulative myelin sheath that wraps and surrounds those neurons. And so this is a very common, it's the most common rare disease. One in about 2,500 people are infected or affected with it. There's over 140 different genes that have been identified to cause CMT. All in all, this affects 3 million people worldwide. And the reason why we're here tonight and I'm talking to you is because there's 0 FDA-approved disease-modifying therapies, and this is what we hope to change. And so one of the most promising, well, there are many, one of the most promising mechanisms to address CMT is through genetic medicines. So let's talk about that. A genetic medicine is a therapy that replaces, silences, or modifies a faulty gene. Every genetic medicine has 2 components. There's a vehicle and the cargo. And this is a theme that we're going to play on as we talk about the different types of genetic medicines. So the vehicle talks about how this is going to deliver the cargo to your body. 2 of the most common ones employed for genetic medicines are something known as an adeno-associated virus, or an AAV. That's this shape right here, or lipid nanoparticles, which is this shape right here. Now, AAVs are incredibly tiny. Lipid nanoparticles can vary in size. I've shown here the scale that they're both about the same, but lipid nanoparticles can range anywhere from being half the size of an AAV to 10 times the size of an AAV. So let's talk about the cargo. What goes inside of these vehicles? Well, it depends a lot on what you want to do. So typically it's a piece of DNA or an RNA, which is why we call this a genetic medicine. And that DNA or RNA is meant to replace, silence, or edit the gene that carries a mutation that's causing disease. And really, it's a strategy that determines the cargo. So the type of genetic medicine that scientists are going to build and invest in depends heavily on what goes wrong. Sometimes you'll have a mutation that's called a loss of function mutation, which that's when your gene is either deleted or has a mutation in it that makes it so that it's not functional. So the protein either isn't made or it's made, but it just doesn't work at all. In that case, the strategy will be to introduce a working copy of the gene. That's similar to the otoferlin gene therapy that we talked about at the beginning, and I'm going to come back to in a minute as an example of how this works. Sometimes you have a gene that is mutated such that you either make too much of it or you can take on a brand new function and make a toxic protein. In those instances, the strategy is going to be to silence or to turn down that harmful message. Sometimes you may want to directly edit or rewrite the code itself. And this is where we could use CRISPR therapeutics, which allow you to fix or selectively disable the mutated copy of the gene. So let's start first with these gene replacement strategies. So these work at the DNA level. And what this is doing is delivering a working copy of the gene using this adeno-associated virus, or an AAV, that I just introduced to you. Now, a little bit of background about an AAV is that an AAV is a naturally occurring virus. We find these in nature, and probably everybody listening to me right now has had an AAV infection at some point. It causes upper respiratory virus. It's a simple cold. It's not typically something that's going to cause a major illness, but will make you feel a little bit stuffy and run down. And the way that this does so is that this capsid here, or this viral shell, has inside of it a single short genome. This genome has these barbell-shaped repeats at the end. They're called ITRs, and that's what gives the genome stability. In these adeno-associated viruses, they carry two genes essential for their function. One is called the cap proteins, and the other is called the replication proteins. Excuse me. And so, in a normal sense and in an effective sense, when these viruses occur in nature, what they do is this capsid fuses with the membrane of your cells, and the DNA ultimately enters the nucleus. And then it takes over that cell's machinery and uses these instructions, these cap and rep instructions, to make thousands of new copies of itself. It bursts that cell open, and then it goes and it finds another cell to infect until your immune system catches up with this process and shuts it down. Now, how do we harness this for good? Well, what we do is we take out that genome, and instead of having that replication and those capsid proteins encoded, we remove them entirely and we put in a therapeutic gene of interest. And so rather than making something that's going to allow that virus to continue to replicate and infect your body, this AAV capsid now is going to drop in a single working copy of the gene of interest, and that gene of interest is going to live in the nucleus of a cell and now make the therapeutic protein that your cell has been lacking. So this is how we can take again, a naturally occurring viruses, and there's many serotypes of these. And depending on the serotype, like for instance, AAV9, they're all given a number. They may preferentially target different tissues, and this is called viral tropism. So in the example of the Otarmeni gene therapy, what the team did was to utilize the AAV1 serotype, which is capable of infecting all of the cells of your inner ear. And we had replaced a working copy of the otoferlin gene into the AAV1 virus. Now, otoferlin was too big to fit in the size inside of just one capsid, so we had to split it across two capsids, which then would come together in the nucleus of the cell. Let me go back for it again. And so in this instance, our vehicle is an AAV1 viral capsid, and our cargo is working as a split working copy of that otoferlin gene. And the way that it works is it infects the hair cells, which are the cells in your ear that receive a sound, and it allows them now to communicate with the neuron sitting below it. Because when patients lack a functional otoferlin, they are unable to communicate with the neuron below, which is why it's called an auditory neuropathy. So the cells are there, they develop normally, the sound can get to the ear, but that hair cell's unable to tell the brain that it just heard a sound by communicating to the neuron. We restore that working copy as we've shown here in this schematic. Now you can record, or you can restore that transmission of sound by firing the neuron to travel to the brain. And so in the recent clinical trial that was completed, 80% of trial participants met the primary hearing endpoint at 24 weeks. So that was reinstatement of sound. And 42% of patients achieved normal hearing, including all the way down to a whisper at the longer term follow-up periods. This is all a single dose, and we expect that a single dose will work for most of that patient's lifetime. So now let me give you an example of how a gene replacement strategy is being used for CMT. This past fall, the CMTRF announced that we were funding a program to manufacture clinical grade gene therapy for FIG4 CMT. So the onset of FIG4 CMT is in childhood. Most of these patients will require a wheelchair and have a very severe manifestation. This therapy, CMT4J, or for CMT4J, had been built by the NIH's bespoke gene therapy program, and during COVID, unfortunately, was shelved for development. There was an opportunity for the CMTRF and Cure CMT4J to come together to fund the manufacturing of clinical grade virus so that the program could then move to development stages and clinical trials sponsored by Alpida Therapeutics. Soon, hopefully, we'll be hearing about the first in human dosing, which is expected any month now. And we are looking to see how this behaves. And we expect, based off of the early studies done in mouse, that we should increase survival as well as motor function in these patients. Either way, a success here will lay the groundwork for an AAV gene therapy across other CMT subtypes, and we'll gain a lot of learnings about how we can safely deliver a gene therapy to CMT patients. Okay, so now let's talk a little bit about how we turn down a gene or how we silence a gene that contains a harmful message. So I mentioned before that sometimes you have that gene that's missing or broken, and we just need to bring back a working copy. Other times you make too much of a gene, and that can create problems, or it has a toxic gain of function property. So in this case, we turn down that message. So I've drawn out our schematic here. Again, you have a gene. That gene is converted into a message. That's the mRNA. And then that message is typically turned into a protein. However, scientists have discovered ways and mechanisms by which you can silence that message. And these are using molecules called ASOs, or antisense oligonucleotides, or siRNA, which are small interfering RNAs. And what these do, these are very tiny, like 19-letter long pieces of an oligonucleotide, which means multiple nucleotides or multiple base pairs of those DNAs, or these siRNAs, and they are very specific to the sequence of that RNA. You can think of it almost like a social security number for a gene, right? So they're social security numbers that are long enough so that everybody can have one that's unique. It's similar with genes of the body. And so you can design these ASOs or siRNAs to specifically knock down a mutant message and reduce levels of protein in the body. So, as I said before, this approach is best when you have too much of something or when the gene is overexpressed or you've got a toxic version. I'm going to give you an example of a SOD1 for inherited ALS, or when you need to silence the broken copy to let the more healthy copy do its job. So the DNA in this approach is left untouched, and the mechanism of action is entirely targeting this messenger sequence that's responsible for building that protein that then has function in the cell. Unlike gene therapies, these don't use a viral capsid. Instead, they can be delivered as what's called a naked, in which case you would do local delivering to the interthecal space around the spinal cord. Or advances in this, which I'll talk about a little bit later, are to coat these ASOs or siRNAs in things like fatty acid ligands or conjugate them to other proteins that act sort of like zip codes to tell them where to go in the body and what cells that they should be dropped into. So now let me give you a couple of examples of how this has been employed. So similar to the gene replacement therapies, I'm going to give you an example that's been used clinically. And in this case, that's called tofersen. So one of the neat things about tofersen is it was developed by one of our Research Advisory Council members, Toby Ferguson. So now you know where the name comes from. And he designed it to silence toxic SOD1. So that was a gain of function copy of the SOD1 gene that causes ALS. And so the problem that was being addressed here is that about some 2% of ALS cases are caused by this mutation of SOD1. You make a toxic misfolded version of it that then aggregates, and these aggregates in the cell are very difficult for motor neurons to handle. And that added burden of these aggregates causes those motor neurons to die. So the theoretic approach to this, which then also showed to have clinical benefit, was, well, if we can turn down that mutant SOD1, we can reduce the aggregates, which would reduce the burden on those motor neurons and allow them to survive. So one of the key results that was observed in this trial was that there's a 35% reduction in toxic SOD1 protein in the cerebral spinal fluid of patients that were treated by 8 weeks post-treatment, and a 50% reduction in a protein called NFL, which is a protein that's released when neurons die. So that's what's called a biomarker. So we're able to understand what's happening inside of the body by looking for this NFL biomarker. When neurons are under stress or they're dying, we see more of this NFL protein released. And so that's why seeing a 50% reduction in that amount of NFL by weeks 12 to 16 was very encouraging. Now let's go back to CMT. So right now in a clinical trial in Canada is a program to silence PMP22. So for those unfamiliar with CMT1A, this is caused by overexpression of the PMP22 gene. Rather than having 2 copies of PMP22 in every cell of their body, patients with CMT1A have 3 copies of PMP22. This represents the most common form of CMT. There's about 150 patients in the US and Europe. As I mentioned, it's caused by this gene duplication. So here, the idea is to reduce the amount of PMP22 being produced into protein by targeting those mRNAs. In this case, this is an example of where there's an innovative therapy, and I'm proud to say that this was initially funded by a CMTRF grant. So this is an example of some of CMTRF's seed funding, where we spent $128,000 to validate that a company called DTX Pharma's Falcon technology, which is a technology that coats those siRNAs or an ASO in what's called a fatty acid ligand. That fatty acid ligand is something that neurons like a lot. And so it allows those siRNAs, rather than going mostly to the liver, instead to not really see much of the liver and instead go elsewhere in the body and be taken up by neuronal cells. This made it so that Schwann cells that are making way too much PMP22 would receive these siRNAs, they're taken in, and then the message is turned down. This program was acquired by Novartis in 2023 and is now in phase 1 trials in Canada, and we are eagerly awaiting results from those trials. Okay, now let's get to our last type of genetic medicine that I'm going to talk about today. So this is what a lot of people, I think of when they hear genetic medicine, which is CRISPR therapeutics. CRISPRs have been very popular in the news, and I'll tell you a little bit about it today. So there's 4 types of CRISPR, 4 variety of CRISPR therapeutics. So there's what you can do, which is CRISPR is like molecular scissors, and it has, similar to those siRNAs, it has what's called a guide RNA. Again, that's like that social security number for a gene that tells it where to go with a high level of specificity. So that guides the CRISPR, which is the cutting molecule, to the right gene where it latches on, and it actually stays on for a very long time, and it cuts. And then after it cuts, it eventually lets go, and now you've got a broken gene. So that can disrupt gene function. So that can be another way to turn down in a way that's permanent because it's actually editing the DNA itself. There's another mechanism that CRISPR can do, which is where it cuts, but then by a mechanism called recombination, you can introduce a healthy copy of a gene into that breakpoint within the genome. That's a little bit trickier and has been more challenging to do within the context of a body, but scientists have been able to do it in cells and culture. There's also something called prime editing. This is another way that we can use a CRISPR molecule, again with that social security number of a guide RNA that directs it to the right place. But instead of making a cut, the CRISPR holds on to it, opens up that DNA, and allows for a new copy of DNA to be introduced that introduces a healthy copy of that gene. That's called prime editing, and something that is in its prime time and being developed by many companies. Lastly, there's CRISPR base editing. Again, this is a type of CRISPR that doesn't make a cut, but has that high specificity by the guide RNA, and instead has this base editing function where it can identify the mutant base and replace it with the correct base. So there's a number of ways that CRISPR can be employed. It sounds a bit sci-fi, but I promise you that it's not. About a year ago, there was a lot of news around a baby that had been born with a very severe form of something called CPS deficiency, which is a metabolic disorder that prevents the liver from clearing ammonia. Without treatment, children born with this have ammonia that builds up to toxic levels within days, and it causes severe brain damage or death. Baby KJ, who we've heard a lot about in the news and in a New York Times article, was too young for a liver transplant, which is the common form of treatment for this disease. The solution was remarkable. A team at CHOP and Penn built a CRISPR base editor that was designed to KJ's exact mutation. They put it in that lipid particle, nanoparticle that I introduced a few slides back as another delivery vehicle. And that LNP went straight to the liver, where it then edited the DNA of those liver cells to restore normal enzyme function. What was really remarkable about this, in addition to just that, here's something that sounds kind of sci-fi and has now saved a baby's life, was the speed with which it was done. It was built rapidly and then got FDA approval rapidly. One of the things that's encouraging for the rare disease community is what's this plausible mechanism pathway that the FDA has laid out. The goal of this pathway is to allow situations like this, where you have a plausible mechanism. In this case, scientists said, we know how we can fix this. We need to use this base editor, and we have a way to deliver it safely with these LNPs. Let us try it. And so that mechanism is hopefully going to open up the door for many other patients born with rare disease where a plausible mechanism exists and help get those therapies to patients. That's something at the Research Foundation that we're keeping a keen eye on to see how we can best apply that in the case of CMT. So now let's talk about the CMT example. So right now for base editors or for any type of CRISPR, there's not an option that's being evaluated clinically. However, there's a great option that was recently published where there's some very good preclinical work that was done in cells that utilizes a CRISPR to cut a dominant negative. So a dominant negative is very similar to a gain of function mutation. What it does is it cuts this dominant negative copy of a gene encoding NEFL, which is a neurofilament protein. And when this protein is mutated, it causes a type of CMT, CMT2E. Since replacement won't work and silencing both copies is a risky endeavor, the goal of these studies was to selectively disable only the mutant copy. And again, using that guide RNA with high specificity, the scientists out of Bruce Conklin and Luke Judge's lab were able to show that they could use a Cas9 with a guide specific to this N98S mutation and selectively cut and disable that gene in the cells that received these editors. So showing that there is in fact a plausible pathway forward for people who have CMT2E with this mechanism. There needs to be additional work to be able to translate these preclinical findings, including the right approach to be able to deliver this type of therapeutic to the peripheral nerve. The reason that we were able to use, we as in the scientific community, were able to use CRISPR therapeutics in the case of the CPS1 deficiency was that LNPs go very well to the liver. The liver takes up a lot in our body. It's the first place things go when they're injected into the bloodstream. But reaching the peripheral nerve represents a much harder challenge. Which takes me to my next slide, which is how are we innovating on these genetic medicines? So really the bottleneck for CMT isn't knowing the right gene. And it's actually not even knowing what to do with that gene because there's been so much knowledge around whether or not something is a loss of function mutation, a gain of function mutation, an overexpression. It's really how do we get this medicine to the peripheral nerve so that if we need to replace, edit, or silence, it can get there. So one way to do that is to engineer those AAV capsids. I told you that they're naturally occurring, but there are ways to manipulate and engineer that capsid shell so that it can be more selective, so that it could cross peripheral nerve barriers, or with a systemic injection, better target Schwann cells or these DRG motor neurons. There's also lipid nanoparticle innovations. So being able to make these tunable lipid-based shells to be non-liver targeting is one way that we can better engineer lipid nanoparticles. There's the fatty acid conjugates that I told you about with the Novartis drug, the EDK-060. This again enriches in peripheral nervous system. And then also looking at redosable platforms. One of the challenges with AAV is that once you've had an AAV delivered, your body has an immune memory of seeing that virus, which is great for us in the normal context of everyday disease when we come across a rhinovirus or we come across an adenovirus or an AAV that makes us sick with a respiratory infection. It's not great when you want to be able to redose a patient with that same AAV. But there's lots of strategies underway to be able to silence the immune system or make these AAVs so that they're cloaked and that the immune system doesn't see it, so that redosing could be possible. Again, sounds science fiction, but it's something that's actively under development by a number of organizations. So the other way we can innovate is to modify the cargo. Again, with CMT, it's not a question of do we know the right gene and do we know what to do with it, but how can we make these genetic payloads smarter? One way is to use a cell-specific promoter. The gene therapy that the team at Regeneron built, we used a built-in switch. This is called a promoter, and it's what controls when and how much of a gene is turned on, when, where, and how much of a gene is turned on in the body. So for that OTAR-many, we used what's called a myosin 15 promoter, which would only allow the otoferlin gene to be turned on in hair cells of the cochlea, which is where it's needed. And it's not because it's not needed elsewhere in the body. We're able to not turn it on elsewhere in the body. The other way is mini genes. Sometimes you have a massive gene, and in that case, scientists are looking at ways that you could remove repetitive elements of those proteins or make mini proteins that still maintain the function, but fit within the packaging size of those viruses. There's also some very neat combinations where you can leverage a silencing approach to turn down a toxic protein and then replace it with a good copy of the protein. So those are called knockdown and replace. And then, as we've already talked about, there's base and prime editing, and that's an area of continued innovation to make that as efficient as possible and to find good delivery mechanisms to get those into the cells of patients. So one of the things I'm very proud of being at the CMTRF is our funding landscape. So we fund a number of genetic medicines and we fund across the board widely. So we're not just focusing on replacement therapies, we're not just focusing on silencing therapies, and we're also not just going with the status quo of what is a good delivery vehicle. We are working with investigators, both in academic and biotech and pharma, to say, how can we better replace silence and innovate on these vehicles? So of our portfolio of 38 funded projects to date, as you can see, there's a number here that are supporting genetic medicines, and we believe that the lessons that we learn from these can be applicable across multiple forms of CMT. So I'll wrap up by saying I think that there is a very bright future for genetic medicines for CMT. And we know that all three modalities have been proven in patients. And even if that wasn't for CMT directly, that initial proof of concept to show that it's possible really can help pave the way for applications within CMT. We know that CMT-specific programs are gaining momentum. I told you about two that are in clinical stages of development. So the ELPIDA FIG4 program. And our next webinar that we'll preview at the end you will hear from Dr. Meghan Waldrop, where she's going to tell you about a gene replacement program for CMT2S. We also have the silencing approach underway by Novartis, and we know that there's a number of other preclinical programs, again, that will hopefully rapidly advance towards clinic. Lastly, as I mentioned before, every program teaches the next. So the more comfort that we get with delivering AAVs, understanding how the body's going to respond, understanding when you want to use a promoter that would turn a gene on in every single cell versus turning it only on in some cells, tells us something about how we can do this safely for the next program. So I'll end by saying thank you. Thank you for your time and attention. Thank you for listening to my story. And as a personal reflection, I would just like to say how excited I am about this. One of the proudest moments that I've had in my scientific career, and one of the most tear-jerking moments, was when I got the video and had heard that baby Opal had her hearing restored. And it's something that people have asked me about. Wow, Meg, that's got to feel really cool. How does that feel? And the answer is, I can't put it into words. Like, the enormity of it is just, it's immense. And so honestly, that's what's brought me here to the CMT RF, is because I hope to translate that exact same work now for CMT. And with that, I'll wrap up and take any questions.
Thank you, Meg. That was incredibly helpful and so informative. Now I'm going to work through some key announcements I mentioned at the top and give you a chance to have a drink of water. And while I do that, I'm going to invite everybody to questions in the Q&A section of the webinar. We're going to do our best to get to all of your questions by the top of the hour, but we will try to wrap up this part of the webinar at 8 o'clock. So we're going to hustle through the announcements and then get to the questions. We also know that was a lot of information, right? Meg makes it look so easy when she describes it. But going into this, because I know it's what I need, I said, can we develop a little cheat sheet that we can hand out to the community? So as you listen to this and you're reading through a research portfolio, you can call to the terms and really start to become your own patient advocate. So in the chat right now, we just dropped a link to a hot off the press, brand new tool I hope is really helpful for you. It's a cheat sheet of terms. They're going to help you to continue to process this information. If you ever have any questions, please just reach out to us. This is the first webinar in a 3-part series on gene therapy. So grab that cheat sheet and bring it with you to each one so you can continue to map all of this complex information as you're learning about what this means for you and your family. All right, so before we get to questions, so pop those in. Here are our exciting announcements. As Meg mentioned, we have another webinar coming. So that's going to occur on June 18th. Dr. Meghan Waldrop is going to come and talk about her gene therapy she's working on for CMT2S. So this topic is gene replacement for CMT2S, challenges in trial design and recruitment. And by attending this webinar, you can sign up for the part 2 right now. So as I'm going through this, links are going to magically drop into the chat. So that link is now into the chat for you. We also have a new project announcement. So just because you're here, we're going to tell you about it first, but this is all going to go out through our channels really soon. But we recently funded a project for CMT2A, and that's led by Dr. Yuri Manor at the UC San Diego. And the full announcement is also in the chat. If you are interested in following 2A research, you can grab that right now and take a look. It's also going to be pushed out to the community. Every year, CMTRF hosts an annual research convention, and this is truly the only research convention dedicated to CMT. This is our 6th annual convention. It's happening September 24th to the 26th in Cambridge, Massachusetts. And we host this event for CMT researchers, patients, families, and supporters. So registration is live. You can sign up now, add this to your calendar. Early bird pricing is available. You can follow the link and check this out. We do highly encourage you to attend in person. It gives you a chance to interact directly with researchers and for researchers to meet patients. But if that's something that's not available for you or the timing doesn't work, you can attend virtually. So please go check it out and add it to your calendar. And finally, your donations from the community are what fund this exact kind of research that you're hearing about tonight. We are hosting a spring campaign that's launching now, and we're actually honoring Alan Jackson through this campaign. Alan Jackson has been public about his very own CMT diagnosis, which we know isn't easy to do, and we're very grateful that he sees the work that the CMT Research Foundation is doing, and he has decided to donate $1 for every ticket for his previous concerts and his final tour for us. And this brings some significant funds to our work. So we've decided to celebrate his legacy by highlighting the number 35. And 35 is the number of #1 hits that he's had in his career. So if you're interested in supporting CMTRF and you're interested in supporting Alan Jackson, there's a link in the chat right now that you can donate directly. When his tour finally concludes, we're actually going to share with Alan directly how much the community raised in his honor. So I welcome you all to participate in that. All right, so I see questions coming in, Meg. I'm going to do my best to get to some here before we get to 8 o'clock. And first, I want to start with a challenging one because there's been a lot of great gene therapy news that we heard about some of that tonight. And congratulations again on the FDA approval. But sometimes we hear about some negative gene therapy news. And that's a real concern. And I know for patients in the community that are really paying attention, it brings some questions to the surface about whether this is a safe and effective path for us. When we see that bad press, what's your honest read on that? And where does your confidence come from when you're in the middle of developing therapies yourself?
That's a fantastic question. So one of the challenges with gene therapy, as we said before, was twofold. It's one, it's the liver, and two, it's the immune system. So where we've seen the biggest successes with gene therapy with the least amount of risk, and I'll put that as it's all relative, right? Because new medicines come with risk. We try as a research community to de-risk as much as possible before going into a person. But when you have systemic delivery of gene therapies, like I said, the first place something goes from your bloodstream is right to the liver. And what the community has found is that in some instances, the liver is taking up too much of that virus. And that can cause challenges both from liver function perspective as well as immune challenges because of just how much is made there in the liver. One of the things that gives me more confidence and more optimism is that there's actually been a number of successes, including the gene therapy for hearing loss, that when you deliver locally, so you deliver directly to the muscle, you deliver to the eye, you deliver to the ear, you deliver to the intrathecal space of the spinal cord. These are somewhat enclosed spaces. You do have some of the virus that can leak out and go to the liver, but it's at a much lower percentage. And this is where the community generally has seen more successes, tends to avoid a strong immune response. And also these innovations using these novel promoters, which are very much within our grasp and which the scientific community has developed, again, add another safety bar to this. And so those are the things that keep me optimistic about it. I think not naively so, but in a way that is you take measured risks and you de-risk as much as possible.
Very good. Thank you. Think the other side of that, Meg, is, obviously seeing the video of Opal and knowing that that therapy, which was an inherited loss of function, and that was just proved forward as safe and effective, it gives a lot of hope in this room to say, is that coming for me? Is that coming for CMT? How directly does what you learn from that program translate to what we're building here? And then a separate question that came in was also, if I have hearing loss with my CMT, is something like that something that will benefit me?
Another two great questions. So I'm going to start with what can translate. So in the answer of the otoferlin gene therapy is that that gene therapy, the otarmeni, won't directly translate to any of the known CMT genes because we're specifically replacing the OTOF gene, which is that gene that's responsible for the auditory neuropathy. However, there are many forms of CMT that come with hearing loss because the auditory nerve is in fact a peripheral nerve. And so when you deliver into that peripheral nerve space, you could expect that some of that gene therapy will reach also your auditory nerve and that you may receive benefit there. It's something that will be tested. There are scientists who have their eye both on CMT as well as hearing loss. Gabriel Corfus of the University of Michigan is one of them. And there are people who are looking at both hearing and vestibular function and seeing if therapies can apply to those inner ear and vestibular functions as well. So I
Awesome.
am
And
hopeful
this
for that.
Awesome. Well, we love when you're hopeful. And this question, I think, kind of morphs from this, but we heard about gene therapy for CMT4J, which CMT Research Foundation helped to develop the manufacturing for that. And so when we hear about progress, and I mentioned a bunch of subtypes at the top here, right, where we're working on it, but we know this is a really complicated disease with a lot of subtypes. So when we're hearing about progress in other subtypes or other genes that are creating the CMT, if it's not the gene we have, what does that mean for us? Like, how should we attach our mindset to that? So when we look at the 4J program, for example, if it's not relevant to us directly, what are the learnings I should be looking for as another CMT patient that's hopeful for a gene therapy?
Yeah, so one of the things that we talked about in the slides was that the biggest challenge right now facing the scientific community and the drug development community for developing genetic medicines for CMT is that delivery challenge. And so these early programs where we're going to deliver, like the Alpida program, where we deliver a working copy of FIG4 for CMT4J, if successful, as we hope it will tell us that we've overcome the delivery challenge. It will tell us, one, hopefully that it's safe, which again, we expect it to be based off of the preclinical studies. And two, if effective, will also tell us that we can in fact reach those tissues that we're hoping to reach, that peripheral nerve, we've done it. We have a way to get there. And so then you can use the genetic knowledge that we have to work on putting in a working copy of a different CMT gene. And now your delivery is further de-risked because now you've seen it go from a mouse into a human and have success there. Same with these siRNA programs. So we're eagerly awaiting the results of the trial for the PMP22 silencing program. That was a DTX Pharma that's now with Novartis. That will tell us if that fatty acid conjugation of an siRNA is reaching the peripheral nerve and enriching it there as expected, and what is the range of doses that we can expect to see therapeutic benefit. This will be a great advancement because typically right now with a naked ASO or a naked siRNA, you have to do that intrathecal injection. And because it's not permanent in the body the way an AAV-based gene therapy is, you have to do repeat injections. Ideally, you move away from having to do repeat injections into that intrathecal space around the spine and instead can go to something systemic, which is what you would have with the fatty acid conjugation on those siRNAs. So again, that's something where we learn a lot about the delivery technology. And when we see that that's effective for one form of CMT, we know so much about the genetics. It's got such a rich genetic landscape with a lot of knowledge there that we know what we do with that cargo. And so it really helps to pave the way just in de-risking those delivery things and seeing it both safe and effective.
The rising tide raises all boats.
That's
right?
right.
We carve the path for one, can open up a path for the others. So thank you for that. We have a family here. It's a good question. I want to broaden it out a little bit, but they recently have their 14-year-old was diagnosed with CMT2E. And they want to know how can they stay close to advancements and potentially participate. So just for that family, I'm just calling that out, but also for everyone on the call, they're here tonight to learn from you. How can they stay in touch with the most recent relevant updates with gene therapy?
Absolutely. It's a great question. So, one is that we'll give a shout out to David, who's often behind the scenes, but is the head of our communications. And David is doing a fantastic job of putting out information and articles as we receive it. We have newsletters, attending our convention, and also just reaching out to CMTRF, letting us know who you are, sharing your email, and we'll stay in touch with you.
Mhm. Awesome. It's perfect. We also, if the team could drop, I'm sure David, you're out there, drop the link to our funded research. We do track this on our website and we do update it regularly. And when big announcements come, we push it out and we'll have a webinar about it. If you ever have any questions about a trial that you see or work that's being done, you could always email us at info@cmtrf.org and we will get back to you and let you know what we know about it. And you might even call to our attention that we'll reach out to that team and start talking to them about their project. All right, Meg. So another really great question. Will adults with CMT benefit from a gene therapy and possibly be able to regain loss of function?
That's a great question. So I don't have a crystal ball. So I can't answer with any level of certainty. But what I can do is I can give you the reasons why I believe I'm not naively optimistic about this. So one is that we know that when we study mouse models and what we see in a mouse model is really that mice aren't humans. And so sometimes we take the information that we get from a preclinical study and we only apply that so far. So let's look then to what we know from human studies. So there's two in particular that I know have a good amount of information on, one being the hearing loss gene therapy, and the other being the recent NMD clinical trial. So if you haven't heard about the NMD clinical trial, I encourage you to go to our website and to look up past webinars because we had one of NMD's uh, employees giving a talk about their program, which is a chloride channel inhibitor that helps to improve muscle strength in CMT patients. One of the things that they saw was that when they were able to apply their chloride channel inhibitor, or when patients took that oral dose, they regained muscle strength. So what that tells you is that even if that nerve is not very functional, there is opportunity to bring back some function. And so even if you're starting to see a loss of something, that tells us that there's potential to regain. Now, in terms of gene replacement therapies, the knowledge that we have is from the Regeneron trial for otarmeni. So this has been published in the New England Journal of Medicine. You can see that there were participants that were older that were dosed with otarmeni. This is something that was a bit of a black box because mice have a 2-year lifespan. So we could look within mice to say, well, the neuron isn't receiving any signal. What you'd expect is that over time, that neuron would die back because it's not receiving input from the cell that's supposed to be signaling to it. What we saw was that older patients who received the drug outside of the window of intervention that initially people thought was feasible indeed responded, which says that yes, that those cells that weren't being used were still there. This gives me hope in terms of CMT because just like the auditory nerve, as neurons begin to die back, it's a pretty slow process, and you feel the functional loss, I believe, before you're actually going to have the complete cellular loss. Again, it's a little bit hand wavy. I don't have a crystal ball. I can't guarantee it, but I think that there are these little bits of information that we can glean that what we expect to see in human is a bit more redundancy than what we would expect from our preclinical models.
Awesome. Thank you. All right. How do you get ready for a clinical trial? So the question is, how do you apply, right? Which might be a little
Yeah.
different, but maybe speaking generally for a clinical trial. And then what happens if the clinical trial is being hosted for your subtype, but it's in a different country?
Sure. Two great questions. The best thing you can do as the answer to both of those questions is to reach out to the sponsoring group or to the clinical trial sites who would put you in touch. Typically, if we talk about NMD, if we talk about Novartis, Regeneron, any pharmaceutical company is going to have an email address that's something along the lines of clinical trials at that, Novartis.com or alpida.org or whatever the organization is. And so I would encourage you to go to their website. If you can't find it, you should reach out to us because we have contacts at most of these organizations and would be able to point you in the right direction of either a person or a clinical coordinator or the info at email where you can get a response to learn more about how would you figure out if you're eligible, how do you enroll, talk to the people who are actually responsible for conducting that clinical trial.
Awesome. It is complicated. And for all clinical trials, I'll just say it's a pretty exclusive list of must-haves, but there's really good reason for that. So if you are ever excluded from a clinical trial, that does not mean you're going to be excluded from the treatment. It just means to really measure true impact for that therapy. There's just some real specificity they need in order to show the efficacy of the treatment. So if you ever wonder about that or feel really defeated because you're not eligible for a trial, you can always reach out to us and we can help you understand it a little bit better. But that's all for the goal to get that therapy approved and through to market as quickly as possible. Oh, we got 3 more questions, Meg. We're trying to do it in 4 minutes, okay? All
Okay.
right. This is a good one. So it's a 2-part question, but can results generated from targeting a single gene or transcript be used to manage CMT in patients where a specific gene is not known? So that's probably more the second part is, what if I don't know my gene? What does this look like for me?
Yeah, so that's where it's a bit challenging. If you don't know the gene, it's difficult to have a genetic medicine that would specifically target your gene. Now, that doesn't mean that you won't be eligible for any genetic medicine, period, because one of the things that scientists are working on now is looking at certain genes that are responsible for promoting neuronal health. And sometimes we want to amplify those genes, and other times you might want to silence those genes if, for instance, that gene's involved in keeping the cell in a very stressed state. Um, so there's programs also that encode neurotrophic, basically is a factor that helps keep a neuron healthy. They can put these into a gene therapy vector. And so in those instances, that form of genetic medicine that's going to bring basically a trophic factor or something that will help keep the nerve healthy, or a silencing factor that might help to reduce or tamper down a stress response in a cell. Those would be examples of genetic medicines that someone without a confirmed genetic diagnosis, but with a peripheral neuropathy, would be eligible for.
Awesome. I feel like you might have said this, but I want to ask it again because it's here. But what would you expect the longevity of an introduced gene therapy would be?
So, with gene therapies, when you go into a cell type that's no longer dividing, so something like a neuron, for instance, the way that gene therapies, those AAV viral genomes are lost is when the cell divides. But our neurons are not dividing cells. And so once they're infected with that AAV, we'd expect extremely long persistence of that viral gene that's been introduced to live within the nucleus of that cell. And so, again, we can only go so far with our preclinical models, and studies in humans that are ongoing are going to tell us a lot about exactly how long we expect these to persist. But indefinitely is the answer because to date, we don't have a known mechanism by which they're silenced.
All right. Awesome, Meg. Thank you so much. Let's see. There's this. I'm going to ask this. I think it's important, but what's your opinion about HDAC6 treatments that seem to target a number of conditions, particularly within CMT?
So an HDAC6, I'm going to start with explaining what an HDAC6 is. I know we have 60 seconds left, but an HDAC6 is a small molecule, so it's different from a genetic medicine. And so it's something that is typically taken orally. These HDAC6 inhibitors are something that help to stabilize that infrastructure that supports the length of these very long neurons that start in our spinal cord and go all the way out into our hands and to our feet. And so my opinion on it is I think we should try as many approaches as possible. And I think that the preclinical data look good. I think also the field is very open to combination therapies. So combining things in the future where you can get benefit with these small molecule inhibitors with something that could be truly disease modifying in the form of a genetic medicine.